So thank you again for joining us, and I will now turn it over to Melissa Thomas, who is Ontario Nature's Nature Reserves ecologist.
Hi, everybody. Thank you for joining. So I'd like to start off with to acknowledge the land on which this project has been undertaken. We respectfully acknowledge that Pelee Island is located in the traditional territory of the Three Fires Confederacy of First Nations, comprised of the Ojibwa, the Odawa, and the Potawatomi. We honor Indigenous communities as the original stewards and caretakers of the air, waters, lands, and resources. In recognition of the Indigenous communities with whom we share these lands and waters, Ontario Nature is seeking to operate in mutual recognition, mutual respect, and shared responsibility. We aim to establish these relationships into the future by operating under the objectives of reconciliation, respect for Indigenous knowledges and cultural practices, conservation of Mother Earth, and sustainable co-governance between the original caretakers of this land and Ontario Nature. So the following presenters will be sharing research and conservation projects that focus on improving and restoring habitat for species at risk snakes, gastropods, salamanders, and bees across Pelee Island. Our host today is my colleague, Smera Sukumar, who will be also presenting with our guest speakers, Thomas Burgess from Trent University, Scott McIver from the University of Toronto Scarborough, Anna-Grant Nicolai from the University of Rennes, France, Anne Yagi from 8 Trees Inc., Jennifer McCarter from Natural Resources Solutions Inc., Jill Crosswaite from the Nature Conservancy of Canada, Thomas Hosey from Trent University, and the last presentation will be delivered by Ryan Wolfe from Natural Resources Solutions Inc., and he will be sharing his findings from his master's research on blue racers. So I'll now pass it on to Smera, who will begin by sharing an overview of this collaborative project and Alvar habitat restoration and management.
Thanks, Melissa. So welcome, everyone. It's so nice to see so many familiar names pop up. I wish we were in a room where I could actually see all of you. So thanks so much for joining us today. So as Melissa mentioned, I work closely with her on the Nature Reserves team at Ontario Nature, and I'll be talking to you a bit about this large collaborative project on habitat restoration on our Stoneyard Alvar Nature Reserve and some of the work across Pelee Island as well. Next slide, please. So a little bit about Ontario Nature. I'm sure you all know we are an environmental charity and we protect wild species and wild spaces through conservation, education, and engagement. Next slide. So we're going to be talking briefly today about the Nature Reserves program. So Ontario Nature has a number of different programs, one of which is this particular one where we oversee and manage 26 nature reserves and 3 conservation easements totaling over 3,100 hectares across Southern Ontario. And you can see where our properties are scattered on this map. And today we'll be focusing on our Stoneyard Alvar Nature Reserve on Pelee Island, which is our southernmost property. Next slide. So as I mentioned, this property is located on Pelee in Lake Erie. All of the projects today that will be shared are taking place across this island. And so this particular property is 42 hectares in size and home to a number of different provincially rare and regionally rare species. And it provides habitat for many species at risk that we'll be sharing with you today. And this property was acquired by the then Federation of Ontario Naturalists back in 1984 as an effort to really preserve these rare ecosystems. So next slide. You might ask what makes a Alvar so rare and what makes them so unique. So many of you may know already that these are natural open areas with a really thin soil profile, so about less than 15 centimeters, and in some areas no soil at all that's covering the limestone and dolostone bedrock. There's barely any tree cover or canopy cover, and definitely not forming a continuous canopy over the area. This photo is taken at our Stone Road Alvar Nature Reserve at sunset, a beautiful time to be there. It's characterized by a variety of distinct flora and fauna, and they're really adapted to some of these extreme weather conditions, such as periodic flooding, drought, and fire. And these disturbances also helped historically with maintaining the open Alvar and ensuring that there isn't too much shrubby encroachment from woody species. And it also helps ensure that it promotes the high diversity of species that are found here. And some of you may know that about 75% of North American Alvars are actually located in Ontario. So we're very lucky here. So if you haven't been on an Alvar yet, I would highly suggest looking up where the closest one is to you, or visiting the Alvars on Pelee Island. And I love this quote from Daniel Brenton that basically says that, on the slide before, that Alvars, anything that can grow on an Alvar is one tough cookie. And I think anyone that knows about the species that live here, and specifically the plants, they're very resilient. Next slide. And so, as I mentioned, there's a number of different disturbances that are a natural component of how these habitats would self-regulate historically, and a major component of that is fire. So this has been suppressed, and so we want to increase natural disturbance in this area, again, to decrease the abundance of woody shrubs and open up the Alvar a bit to increase the abundance of the native Alvar species that we find on these lands. Next slide. So as you can see here, Ontario Nature's Stone Road Alvar is in the red border. We're surrounded by other conservation lands, including lands owned by Nature Conservancy of Canada and Essex Region Conservation Authority. So it is a Stone Road Alvar complex, and our parcel is just one part of that complex. So we work closely with conservation partners to ensure co-management. And so our purpose of the Prescribed Burn Project was to restore the Alvar habitat. There's two distinct grassland Alvar habitats found on this property, shown in green in this slide. And we also wanted to determine the effectiveness of prescribed burning as a stewardship action for maintaining key habitat for these species at risk. This project started in 2017, and monitoring started in 2018. So we did 2 years of pre-burn monitoring and 2 to 3 years of post-burn. Unfortunately, there were some gaps in data in 2020 due to the COVID-19 pandemic. And we've been monitoring a number of different species, and some of those our speakers will talk about later today. Next slide. So now Melissa's going to share a short video. It's kind of a time lapse of some photos from the actual prescribed burn. You can press play whenever you're ready, Melissa. And this basically just shows the lighting of the prescribed burn, the fire. Lands and Forest Consulting were employed to carry out this low intensity prescribed burn in September 2019. As you may see, it's really low intensity. So it's like shin to knee height. And I don't know if you notice when looking at it, you can go to the next slide. It's really low intensity, kind of, carrying through as much as maybe we had hoped. The wind was really low. It was kind of a patchy burn, and you can see some photos after as well. So there's definitely scorching of the ground, but unfortunately, the weather conditions didn't permit for as high an intensity burn as you were hoping, even though this was a low intensity burn. So next slide. So in terms of some lessons learned, prescribed burns on Pelee are logistically challenging. Burns anywhere are logistically challenging, but even so, more so on Pelee, especially with the changing weather. So As I mentioned, it was a bit lower intensity than we had planned due to increased moisture on the ground than we were expecting and lower winds that weren't able to carry the fire through. This achieved a really low intensity patchy burn. So some areas that were scorched, some areas that were left open. We did want a patchy burn to enable refugia for species to get to, so that they could seek shelter safely. But it was a bit more patchier than we were expecting. In terms of the impact on the woody species that we were hoping to push back, it didn't have as large an effect. And so in terms of next steps, we want to continue with our active management to maintain the open Alvar habitat on this landscape. And now we're going to be moving forward with mechanical shrub removal, which will be left on site. The shrubs will help with increasing the fuel load and carrying that burn through. And of course, this is funding dependent. And one thing that, we're really short on time today, but I did want to acknowledge that, conservation partners have enabled us to continue some of this monitoring, not just on Ontario Nature's lands, but on lands across the island. And so we have some control sites as well as this burn site, and have been testing mechanical shrub removal on a number of different sites as well. So these next steps are really based off of monitoring and research that have been taking place since 2018. So I'll pass it back to you, Melissa. Thanks.
Thank you, Smera. So I'm going to pass it off to Thomas Burgess to talk about small mammal community response to the prescribed burns on Alvars.
All right, thank you, Melissa. Hello, everyone. I'm Thomas, and yeah, I'm going to be talking to you about today the small mammal response to the prescribed burn in the grassland Alvar. So next slide. So the primary thing we were looking at was how will restoring grassland Alvar affect deer mice, the primary small mammal species on Pelee Island. So there's two different restoration techniques that were used, as was described earlier, the prescribed burn and the mechanical removal. And what we were expecting to see from both of these was the prescribed burn and the mechanical removal were both supposed to help increase the abundance of deer mice on the Pelee Island Stone Road Alvar. And this is because the prescribed burn removes all the vegetation and drops the seeds from all the vegetation, which the deer mice really like. And they tend to move into areas after prescribed burns and just after burns in general to go after these seeds. And the mechanical removal is supposed to help the deer mice because it provides the refugia that deer mice like, where they can hide in, they can get some cover, and these little guys really like that stuff. So we had 3 different monitoring sites when we were examining this, and that was the prescribed burn, the mechanical removal, and just a natural habitat. So this was an area that we had no different treatments done at it, and this was just supposed to give us a sense of what the mice populations were doing when we examined the mice populations with the prescribed burn and with the mechanical removal. Next slide. So what was done? There's monitoring done for the small mammals in the spring from 2019 to 2022, small gap in 2020, just thanks to the pandemic, which we all know and love, right? But we had treatments done in the fall of 2019. So when we discussed about trapping them, we have this grid that you can see here. It was a grid that was set up as 5 traps by 10 traps for a total of 50 traps set. This was checked twice daily for about a week in the spring, whenever we had the 3 different treatment sites. And this is what we use to catch and see the abundance of the mice in the area, as well as like the trap success and what they're looking for. And once we caught a mouse, it was tagged, we recorded some information, and then it was set free back to go run around with his buddies. So at the different treatment locations, we also did some vegetation sampling to take a look at the effect of the different treatments and just the natural habitat, see what's going on, and see how the mice were responding to all these, everything going on in its natural habitat. Next slide. So what we can see here is that the mice population over time did in fact have a decrease from 2019 to 2022, but it's important to note that it was across all 3 different treatment sites. So we didn't really have, it's hard to really tell from this directly if there was a negative or a positive effect from any of the treatments, because even in the natural habitat where we did nothing, there's no real anthropogenic or human interference there, that the mice population did decrease. It is interesting to note, though, that in 2021, from 2019, at the mechanical removal site, we did have an increase in population, before it eventually did decrease in 2022. So what we're seeing is over time from 2019 to 2022, that there's a decrease in the mouse abundance. Next slide. So what can we use to explain this? What explains the change in mice abundance? Now, this table here is just showing a bunch of different models. The key takeaway from this table is just that top corner right there, model, year, and treatment. What that's showing right there, that it is the effect of both year and the treatment on the mice populations. So this is showing that two different things. Year is having an effect, so there is a variable effect in the mice population across the different years, but it's also showing that there is an effect in the treatment between the different treatment zones. So the prescribed burn and the mechanical removal both had an effect on mice populations. Next slide. So by examining now that we see that the treatment had an effect, as well as the year had effect, how can we break this down more? We were looking at trying to find specifically what was going on. And what we found was that the mice very much like the coarse vegetation, the woody debris. And that's found with this table. Again, the only thing you need to know from this table, the key takeaway are those top three things. Mice like woody debris nearby. So the bigger it is, the more decayed it is, and the amount there is around it, they like that. So the more of that you have in a site, the more the mice will find it. More mice will be there. Next slide. So the change in vegetation over time, you can clearly see that there might have been some increases, but overall it all has a decrease from 2019 to 2022, which is very similar to what we're seeing with the change in mice abundance over time from 2019 to 2022. So it's interesting to see that even though all these different treatments had a different effect, because you can see none of these three graphs are none of the graphs are the same across year or treatment. We're seeing that there is a decrease in vegetation from 2019 to 2022, and that's what's driving the mice response there, the fact that they are decreasing from 2019 to 2022. Next slide. So the key takeaway here is that we definitely had treatment had an effect on the small mammal population. And this effect on the small mammal population is what we're seeing with the changes in mice abundance. But more work is needed to be done to fully understand this response, because we saw mice population and vegetation decrease even in the control, which we had no effect over. So more work is needed to be done to see if the prescribed burn had less of an effect than just what was going on naturally in the mice's natural environment and just across time. That's all I have for you guys today. Thank you for listening.
Thank you so much, Thomas. I'll now pass it on to Scott McIver, who is going to talk about the impacts of prescribed burn on wild bees and flower visits.
Great, thank you. Thanks everyone for attending, really good turnout. Today I'm going to talk about some of the research we've been doing on Pelee Island, looking at the effect of the prescribed burn on wild bees and the flowers that they visit. This is work that we do at the University of Toronto Scarborough and in conjunction with Dr. Mark Haddad, who couldn't be here today, but I won't be speaking much on the plant side of the research, but much of our work is in collaboration, and I'll hint at that at the very end, where I introduce our new PhD student, Janine Sharkey, who will be conducting much of the research moving forward in this realm on this project on Pelee Island. So here's just a few of the bee species that we see here on Pelee Island. Let's do the next slide. So first and foremost, we don't know a lot about the bee diversity on Pelee Island. We anticipate that it is rich. There's a lot of unique habitats and floral specialists that we would find on Pelee Island. Of course, because it is the southernmost tip in Canada, we also expect new records for Canada. And additionally, because it is a fairly unexplored bee fauna, it is possible that we may find new, again, records for Canada, potentially even new species. So an important part of our research fundamentally is to develop a bit of a regional checklist of the bee diversity that we find in the Stone Road Alvar on Pelee Island. And so to do this, we've been working consistently across 6 sites, three different sites where there's 2 different survey areas. We've been using vein traps to survey before and after the burn. As we heard, COVID kind of has dampened some of our ability to sample on Pelee for a little bit of 2020 as well as 2021, but we'll be back in full force this coming year. Next slide. And the goal in collecting this regional checklist is, of course, one, to have a baseline, an understanding of all the different species that we find there, so we can begin to evaluate particular patterns relating to management practices, invasive species. And this collection will be curated and housed at the University of Toronto Scarborough in our bugs research collection. Hope to host at the museum on Pelee Island. A synoptic collection is an example of a male and a female of all the species that we find on Pelee. This will allow us to do further work on looking at abundances and different trait states that might be important to identify as under threat or important for surviving things like a burn. Next slide. Additionally, of course, with the floral richness we find on the Alvar and on Pelee in general, an important part of this work is to identify the flowers that are important for supporting bees that we deem to be of conservation concern. Similarly, many of these flowers may be under threat, and understanding a little bit more about the pollinators that visit them may allow us to create pollinator habitat that support these flowers that may be of conservation concern. So we're also netting flowers, netting bees on flowers across these environments as well. Next slide. And from the bees that we collect, we can remove the pollen from their bodies. Different bees and flies, in this example, harbor pollen in different locations. We can remove this pollen, identify it in the lab, look at the diversity of those pollen grains to infer the breadth of species that they're visiting in a single foraging bout. We may be able to do work on heterospecific pollen transfer and pollination syndromes and pollen limitation and more delicate experiments in the years to come as well. Next slide. And additionally, we're interested in taking the information we know about the bees and the flowers that they visit to construct these networks. And these networks can allow us to make inferences about the stability or the resiliency of particular habitats in terms of the plant-pollinator interactions. We can compare these across the different sites and before and after the burn. Next slide. And that's fundamentally what we hope to be able to do once we have finally able to pull this collection together, is assess the diversity before and after burns. What we might expect is that pre-burn, there's certain non-native invasive species that are highly attractive to bees, and they become a bit of magnets to the populations, ultimately filtering the community towards those that like non-native species more. When those species are impacted by the burn, allowing native species to survive and persist in these environments, that may greatly strengthen this network, adding more capacity for specialist species, for example, that might be of conservation concern. Next slide. We also might expect that different functional traits would be important. For example, cavity nesting bees, like those that nest in stems. If we conduct a burn, we might see a decline in their populations through time, but we may see an increase in the presence of ground nesting bees once the vegetation is cleared, that opens up space for them to nest. Next slide. And finally, this might reflect different functional trait spaces, which may be important, again, in the context of supporting the conservation of community level indices, right? Bees are diverse, so are flowers. And if we can make sense of these broad scale patterns, that might be important for conservation too. Next slide. So just quickly, two very common bees which we found are Osmia conjuncta, which is a snail shell nesting mason bee, the only one that we have in Ontario, as well as Bombus griseocolus, the brown belted bumblebee, which is a very common bee, doing very, very well on Pelee Island. Next slide. And additionally, you can see very quickly, just running through this, that there are some groups that are very overrepresented. Ceratina, these are small carpenter bees. They nest in stems. So for example, on the EC site, this is very woody, very dense woody site where there'll be lots of spaces for those cavity nesting bees to nest, and we see that. And conversely, on the Sunward Alva, which is quite open, but littered with snail shells, this provides more habitat for those mason bees, Osmia, that prefer to nest in those shells. We're still working on a lot of these data. This is some very preliminary stuff. Next slide. And so we have, again, collected all of our samples together, and we're in the process of identifying them. And our new PhD student, Janine, will be doing a lot of that work. Next slide. Oh, one more. Yep, there we go. And we are in that process now. So anything really tough, there's some groups, Lasioglossum and some of our kleptoparasites that we found on the island that we need to do DNA barcoding on. And we will be doing that at the Canadian Museum of Nature in Ottawa. And we'll continue to collaborate with Marc Haddad on the plant side, starting to bridge these two communities together to make some important implementation advice and best practice for this particular environment. Next slide. And just to end, and I think we'll go nicely into the next set of presentation, is an introduction to our new PhD student, Janine Sharkey, who's co-supervised by both Marc Haddad and I. And she will be pulling these two data sets together we've collected over the last few years, as well as embarking on a new set of experiments to link plant, pollinator, and below ground properties together. So as, for example, the burn impacts the plant community, it also is influencing the snails and the earthworms, and this might have influence on the plant growth and traits, as well as floral properties, which may influence pollination success. Starting to link all these pieces together. Janine has experience with bee identification all through the Ojibwe Prairie, so she's very well experienced in working with these data set, and we're really excited to share some big new updates next year. Thank you very much.
Thank you so much, Scott. So next we'll move to Annegret, who will talk about the impact of prescribed burn on endangered snail and invasive earthworms.
Yes, so hello everyone, and thank you for the invitation to this webinar. Next slide. So snails and slugs and also earthworms are important members of the soil ecosystem. Next, especially in the Alvar, where the leaves, the dead leaves from the plants that fall on the ground, next will be decomposed by the topsoil footweb, and then this gives back the nutrients to the plants. So next, snails and slugs. So this time we also worked on slugs, and earthworms are the primary decomposer in this topsoil footweb. Next. We have a lot of endangered species of snails and also one slug species on Pelee Island. And these species really rely on the leaf litter for food, as we see, because they are the primary composers of this leaf litter, but also for shelter because it gives specific microclimate to them. And our research question was, will the fire negatively impact the species on Stonewood Alvar? Because we suppose that the fire will destroy the leaf litter. We also have a lot of invasive species, earthworm species on the Alvar, so we don't have any native earthworm on Pelee Island, only European ones. And these primarily feed also on the leaf litter, and so they mainly destroy the snails or other primary decomposers. So they destroy the snails' food resources and the shelter, and our research question was, will the fire help to control these earthworms? Next. So first we worked for the several last years on broad-banded forest snail, which is a snail that really lives on the Alvar and Stonewood. Next. And we could, during the last work, last years from 2017 to 2019, we could identify some zones with different densities and specifically in the burn area. So you can see in the northern part, it's a zone of a higher density, while the other parts have less high density. Next. And after the burn in 2020, we saw that in the southern part, the density stayed the same. In the middle part and central part, we had no snails at all. And in the part where we had a high density before the burn, after the burn, the density was quite low. When we came back this year in 2020, next 22, so the density from the part where the density was high before the burn came back to what it was before. So this means that the snail very quickly recolonized this area. Next, we also looked at other species at risk. For example, striped whitelip, which is a species that lives more on wet forests. Then eastern banded tiger snail, which like forest habitats, but with loose stone on the ground. And the last one, the Carolinian mantle slug, which is a typical forest snail, which really needs old rotten wood, so logs on the ground. Next. So we looked at these species in the entire nature area, and you can see that for example, Carolinian mantle slug is not concerned by the burn because it lives really outside of the burn area in the more forested area. The eastern banded tiger snail, which also prefers more forest area, is on the edge of the burn area, and we couldn't see any changes in density between before and after the burn. So this means it was not affected by the burn. Interesting is the striped whitelip, which is quite in the burn area, but there was also no changes in density that we could have noticed. Now for the invasive earthworms, you should know that we have different functional groups. There are the EPG group, which really lives in the leaf litter on the ground and only feeds on the leaf litter. Then we have the EP-NEC group, which is in the topsoil. It burrows slightly into the soil, but mainly feeds on the leaf litter. Then the strict NEC, which is also in the topsoil, it can go deeper in deeper horizons, but the same, mainly feeds on leaf litter. And then the last group is the endogeia group, which is only in deeper soil horizons, and it only feeds on organic matter, which is already in the soil. So we compared the earthworm communities in two areas, in the burn area and in the control area, which is the NCC property. And next. And we saw that there's a difference between the abundance of earthworms in Ontario Nature Property and in NCC, and this is mainly due to the soil depth. So on the Alvar, there's the soil is less deep, so we have less species. Then we could know, please come back. From 2019, so from the pre-burn situation to 2020, you can see that the abundance decreased in the two sites. So this means it was not caused by the burn because it happened in the control site as well as in the burn site. So we thought it was more related to climatic conditions, which we can typically observe in earthworms, that when the summer is very dry, then the earthworm community goes down. So the number of the members. So next. What is interesting is that in the Ontario Nature Property, so where the burn happened, that the community has changed. So before the burn, the community was composed of the endogeic species, but also of EPG that feeds on a leaf litter. After the burn, these species completely disappeared. And 3 years after the burn, some of the EP-NEC species already come back. And on the NCC property, we can see that the community composition has not changed at all. So here we can really see that the burn had an effect on the species composition of the earthworm community. So next, so in summary, next. In response to our research question, if the fire has negatively impacted the snails and slugs on stone at Alvar, yes, for some species that live in the burn area, but recolonization was quite fast, so within 3 years. For the earthworm species, we can see that, or for the earthworm community, that there was an effect in the first time, so very short term, but the community re-established very quickly. Within 3 years, and it will probably continue. So that's what we will see next. For the next something that we will do in the next year, we will see if they really fully establish. And as Scott already said, we are also involved now in some work that relates so this below ground biodiversity with the above ground biodiversity, which means plants and bees. Thank you very much.
Thank you, Annegret. We'll now move on to Anne Yagi, who will be talking about sharing her work on snake hibernacula on Pelee Island.
Hello, thank you for the invitation for coming. We are monitoring and building hibernation habitat on Pelee Island, and we began our research in 2018. And in order to do this research and to build and construct, we had to study the unique subterranean environment of Pelee Island during winter. Next. So one of the key areas of study is that upper fractured limestone area that forms caves and cracks and crevices. And we've been monitoring groundwater levels in a 4-meter deep well that we found in the Alvar area. And we found that Pelee Island has a very unique system with respect to groundwater, and which they have a unique pumping system, of course, which is necessary to maintain the island community. And the microtopography on the sites that we are looking at are quite flat, and most are dry most of the year. The surface soils are silty clay and quite mobile. So all these factors we had to put together to design our artificial hibernacula for Pelee. Next. So our goal is to investigate and characterize the subterranean environment for snake hibernation habitat suitability, and really to build new sites and structures for snakes. Next. So some of the facts about snakes and winter survival that we've been researching, I've been researching for over 25 years, is that, but some basic understanding is needed that, snakes are ectotherms, and they use behavioral thermal regulation to seek out optimal thermal areas to meet their life cycle needs at the time. They don't eat during winter. Okay, and they spend over 200 days underground in some places in Canada. And in Pelee Island, maybe because it's warmer, it's a little bit less, 150 to 180 days is standard. They also can't survive winter in a frozen state. Okay, they are good divers and they're naturally good breath holders, but they need access to air oxygen. That is what my research has shown, to meet their metabolic needs, even at cold temperatures. Next. So some of the overwintering threats I'd like to talk about. We know that snakes try to hibernate in the shallow overburden areas in any holes that they can find on the surface. And this is likely a really poor strategy, as some of these areas are not very deep and they're prone to freezing and flooding during hibernation. Here's a picture from Ryan Wolfe of a blue racer that tried to hibernate in a broken drain hole on the surface of the ground, and it died from flooding that winter. So these drain holes are basically ecological traps that we need to look at in the bigger picture of things, and maybe have a capping program for that. Next. So some of our research, and I won't go into too much details, you can go online to see some of what we're doing, but we know that body condition of snakes is really important before they enter winter to improve their survival. But most importantly, the habitat must maintain a life zone, which is a subterranean space that does not freeze or flood completely and remains aerobic and meets their oxygen needs for winter. Okay, so our designs provide a stable water level independent of what goes on the island. And our hibernaculars therefore have a static controlled level, and that provides thermal mass and stability over winter, humidity and hydration needs of snakes. So our research, after controlling the water levels, our research is now focusing on the effect of overburden and the depth of overburden on maintaining the internal temperatures of our hibernacula as we move forward. Next. So we've been studying a 4-meter deep well, and this is a thermal profile of that well. The really variable lines are the air temperature, and you can see the more stable lines are the ones at depth. The dark blue line on this picture is the 4-meter depth, and it maintains during winter a temperature of between 8 and 10 degrees, which is what we expect to see. And about 1 meter depth from the surface, that's the yellow line, it maintains temperatures at 4 degrees C, and that becomes our target for the construction of our hibernacula. Next. So we've also looked at groundwater levels in this hole, and we've been able to show that pumping on the island really stops or slows down quite a bit during winter. And the variable lines there that you see, the very sharp peaks are when surface water gets into our well, because our well is not capped. But the groundwater pumping really begins in May. And the really crazy lines more to the right of that graph are the effects of pump testing on the island. Next. So our structures are built isolated from the Pelee Island pumping system, as I mentioned, and we have a constant controlled water level within the structures to provide that thermal mass. So we build above ground, and microtopography is really important, but the variable that we're really focused on over the next little bit is the amount of overburden that we have around these structures. Next. Inside our structures are really clever designs of interior spaces to allow snakes to move vertically up and down. And we are monitoring 3 large ones that we just built in November. So they're new, they're 100-gallon sized cisterns. And then and we're monitoring 3 older ones that we built in 2020 that are 25-gallon cisterns. Next. And really, can't stress enough the importance of monitoring the life zone within these constructed hibernacula over consecutive years before allowing snakes access to anything that you may build. On our experience, we aim for 4 degrees Celsius as the minimum temperature. And right now, some of our constructed sites are a little colder. They get down to 2 degrees, and I would like to see that improved. So we are working on that. So anything really less than 4 degrees Celsius in the internal chambers is really risky for snakes in their survival. So even on Pelee Island, which you would think would be our southernmost place, right? But we have really harsh winters. We don't get a consistent snow cover, and there's very little overburden, which helps protect these chambers. So so they're very vulnerable to the polar vortices that we're seeing and with climate change effects. And that's something that we're keeping an eye on. Next. So finally, I'd like to leave you with an opportunity to see the world through a snake's eyes a little bit more. This is through a lens of a thermal camera. And to the right is the image of an entrance hole that we have at our constructed hibernacula. This was taken during freezing surface temperatures, air temperatures of -5, which is the blue color in the thermal image. And then you can see the chambers are showing a warmer entry hole, which means that would attract snakes inside. Next. And this is looking down into our chambers on a very cold day of -3 degrees, looking down our monitoring well into the chambers at 7 degrees Celsius in the water and -3 in the air temperatures. Next. Thank you.
Thank you, Anne. I'll be passing it to Jen to talk about blue racer nesting habitat on Pelee Island.
Thank you. Hi, everyone. Thanks so much for coming. So I'm going to talk about a little bit about nesting habitat and what we're doing to help create nesting habitat on Pelee Island. Next. So I thought first I would talk to everybody about what blue racers are nesting in naturally. So blue racers do lay eggs, and they're seeking out areas on the landscape that would be suitable for incubating those eggs. So they're looking for things like rotten logs, cavities underneath the large rocks, compost piles, etc. Anything that will get those eggs, that will protect them and get them up to temperature for incubation. So these sites also have to be in sort of open canopy, sunny areas, as you know, the sun in conjunction with the rotting material helps to create warmer areas to incubate those eggs. And a previous study done on Pelee Island suggested that nesting habitat is limiting on Pelee Island. So we wanted to investigate this a little bit further. Next. So in 2019, we looked at random plots throughout the blue racers range on Pelee Island in areas that we had already identified as potentially suitable nesting habitat. And what we found was that the majority of these plots either had no suitable nesting structures, so no discernible logs or tree stumps or rocks. So that's like the picture on the right. So it's open canopy, but there's no real structure for them to nest in. Or the majority of our plots that had suitable nesting structures like logs, like stumps, etc., had closed canopies, so no sunlight, and these are probably going to be not reaching the temperatures needed for suitable incubation. So that's like the photo on the left. This site had a nice big rotten log, but was pretty well fully covered by the tree canopy. Next. So with those results in mind, we thought, yeah, okay, it does look like nesting structures are limiting for these snakes on the landscape. So we set about trying to enhance nesting habitat for them. So we have built a total of 30 nesting structures. We built 10 in 2019 and then another 20 in 2020. And these are placed throughout the blue racers range in areas that are in or adjacent to areas that they're actively using, as well as an open canopy or southern facing areas. The structures consist of page wire fencing that's constructed in a cylinder and then filled with nesting material such as locally sourced straw, leaves, wood mulch, as well as sticks to try and create little gaps for snakes to use throughout the nesting material. So we were working as well with a colleague of mine who was also constructing these structures in Norfolk County. And so together we're testing 4 different treatments. 2 treatments are just 100% straw with 2 different amounts of woody debris throughout the straw. And then the other 2 treatments are a triple mix of the straw, leaves, and wood mulch. And again, with the 2 different amounts of woody material to try to figure out, is one of these mixtures better for nesting than the other? So we'll try to see that. Next. So since they were constructed, we have been monitoring these structures for use. So the first 10 structures that were constructed were monitored in 2020. And then in 2021 and 2022, we've gone back and monitored all 30 structures. So this consists of taking the Pageoir fencing structure off and then sorting through all the material on a big tarp. So hand sorting through really carefully, looking to see if there's any evidence of nesting by blue racers in these structures. So looking for snake sheds, but primarily looking for eggshell fragments that would indicate that they had been used for nesting. In addition, we've also been putting in temperature data loggers to look at whether or not there's suitable conditions for incubation in our structures. And we've been also looking at moisture levels in the structures. Next. So unfortunately, to date, we still have no evidence of any nesting by blue racers in these structures. But we have been finding lots of other wildlife using the structures. So just some photos here of the different things we've been finding. In the top right corner, there's a picture of a rare horned Parnassus beetle that we just started finding this year. So that was really exciting. And we also had lots of salamanders using the structures because the inside of them gets quite moist with the material breaking down. So lots of salamanders using them, lots of toads and other snakes just thermoregulating or seeking shelter in the structures as well. So while it is disappointing, we still have funding for 2 more years to keep maintaining and monitoring the structures. So, fingers crossed, we're really hopeful that as the structures have been out there for a few years now, that they will be found by mature females and hopefully we'll start to see some nesting in them soon. So stay tuned. Thank you.
Thank you, Jen. So I'll pass it on to Jill now to talk about the Nature Conservancy of Canada.
Thanks, Melissa. Hi, everyone. I'm going to give a brief overview of what the Nature Conservancy of Canada is and some of the work that we're doing on Pelee Island and talking a little bit about the conservation context of the island as well. So next. So the Nature Conservancy of Canada is a nonprofit, non-government land conservation organization. We work across the country and have protected with our partners, more than 15 million hectares. And that includes more than 196,000 hectares in Ontario. And we prioritize where we work based on the importance of these areas for habitats and species, and have been around since 1962. So next. So Pelee Island is one of those areas that has really popped out as being a priority for conservation. As Smera was talking about, it's full of rare species and rare habitats. So it's really important to be looking at that for protection in the future. And it's at this point is now a really unique situation in Southern Ontario where 18% of the island is formally protected for conservation. So 10% of that is owned by NCC. You can see the map on the left identifies the areas that are protected for conservation, and the other 8% is protected by Ontario Parks, Ontario Nature, and Essex Region Conservation Authority. Next. So because there are so many rare species, they have different needs, as they've already been talked about. Some need forests, some need moist conditions, some need open, warm conditions. So it's important for us to be balancing our habitat management and making sure that we are providing the habitat all these different species need. and understanding as well how these species are responding to different management, because so much of the island is protected for conservation. It's this really unique opportunity to be able to ensure that we've got the habitat we need to have stable populations of all of these rare species. Obviously, collaborative management is key, and knowing how these populations, their current state and how they're changing over time and how they're responding to management is critical. And that's why all of these research projects are so important for us as conservation land managers to be able to know what we need to do for them. So we've talked a lot about Alvar. I'm going to shift a little bit and talk about some other habitat types that are important on the island and what NCC has been doing there. Next. So wetland restoration is something that we've been working towards recently on Pelee Island. So obviously wetlands are playing a lot of key roles in water retention, filtration, flood reduction, drought reduction, as well as being habitat for a lot of rare species. So some resident species and as well as migrating birds. So Pelee Island is a spot where it's a stopover habitat for a lot of birds as they're migrating north and south. And wetlands are one of those places where a lot of birds will stop to come down and refuel as they're migrating. So Pelee, back in the 1800s, prior to, it was mostly marsh actually. So it's a series of islands connected by marsh. In the late 1800s, it was drained and that land claimed for farmland. So a lot of that marsh habitat is no longer present on the island. So one of our key goals has been to reestablish that where it's appropriate on our lands. So the red circle on that map on the left, the map's showing where the marsh used to be. The green spots are the NCC properties, and the red circle is the area that I'm about to talk about next, where you can see that there's an island as well as some marsh habitat. So next. So over the past couple of years, we've been working to establish a 25-hectare marsh back on that former marsh habitat. So this used to be a soybean field. And we started off in 2020, you can see in that top right picture there, that's October 2020 where we're working on construction. So some deeper areas were excavated in the field and that material used to build a berm around the outside of the field. And all of the farm tiles, so these drains that run through the fields that pull water off quickly, those were all broken as well to help retain water in the landscape. So the picture on the bottom right is actually May 2022 in pretty much the same spot, looking at the same angle. So the construction was really successful. We've already started to build up marsh habitat in that area. A lot of rare plants have showed up. A lot of birds are using these areas already. We've seen thousands of ducks and shorebirds. We've got rare plants, including Scarlet Amania, which is the one that you're seeing on the background on the right. And these have all appeared. A lot of the plants are showing up without our help. They're already in the seed bank and they're already ready to go once we put that wetland habitat back on. So another really cool opportunity with this is recognizing that this is habitat for species, but humans are also a species and they need places to go and be in nature. And this is one of the places where we've had an opportunity to really build what our visitor infrastructure opportunities for hiking and bird watching. So next slide. So because this is new habitat, we don't have to do anything that's going to impact existing habitat. We're not removing anything to create trails and blinds. So we've built that right into the restoration of this area. So this year we've really focused on building that infrastructure. We've started on building a blind that will allow people to come and look out over the marsh, watch birds and other wildlife. And this is fully accessible. It's got a ramp that leads up to it, so everyone will be able to get to it. There's a few finishing touches, but it will be fully complete in spring 2023. And we've also created a 2-kilometer trail around the outside of the wetland as well, so people can see it from different areas. And it connects into the trail network elsewhere on the island as well. So it's really a great opportunity. People will be able to learn more about the rare habitats on the island, the restoration that's happened, and be able to get out in nature a little bit more. And I hope that many of you, if you haven't been there already, will take the opportunity to go visit. Thank you very much.
Thank you, Jill. So I'll now pass it on to Thomas, who will talk about salamanders and the unique salamanders on Pelee Island.
Okay, thanks everyone. Yeah, so I'm going to give you a brief overview of the work that we've been doing to study the unique salamander complex on Pelee Island. So next slide, please. So as you can see on the map on the left, we have the distribution of blue-spotted salamanders and smallmouth salamanders. And Pelee Island is located basically in one of the few places in the world where these two species overlap. Can you show the next animation, please? So there's Pelee Island. You can see that it's one of the few places where these two species co-occur, and it's the only place in Canada where the smallmouth salamanders occur. There's also a third salamander called the unisexual salamanders, and they live alongside these salamanders as well. And they have DNA from both of these species, and they're reproductively dependent on sperm from them. One of the challenges for us, though, is that they can't really be, you can't tell them apart from smallmouth salamanders and blue-spotted salamanders just by looking at them. So we need to use genetic tools to help us distinguish who's who when we're doing our research. So as we heard from Jill, Pelee Island historically was largely a marshy wetland. A big part of Pelee Island was wetland habitat. And when that was drained in the late 1800s for conversion to agriculture, the remaining habitat for salamanders was kind of fragmented and diminished. So the salamander populations today are sort of fragmented and scattered across the island. Next slide, please. And so our work began in 2015, and we've been studying various aspects of the salamander ecology. And our work has involved the discovery of new populations, genetically characterizing the composition of these populations, sampling at different parts of their life history during the adult breeding stage, the larval stage, trying to get a handle on population size, engaging in a mark recapture study, looking at habitat connectivity and habitat suitability across the island. And doing a variety of other things like just characterizing the breeding habitat, what makes a good salamander pond, something we'll talk about at the end, and evaluating and comparing constructed ponds to natural ponds to see if the constructed ponds that exist on the island are providing good enough habitat for the salamanders. In addition, we've been able to use all of the data we've collected to help conservation organizations identify areas for restoration and habitat creation. Next slide, please. So as I mentioned, we need to use genetics to understand the genetic composition of these populations and also just understand how they might have changed. And so here's some historical data from 1984 to 1991. And the LL columns and the TT columns, LL is for the blue-spotted salamander, the TT is the smallmouth salamander, and the LT combinations are different types of these unisexual salamanders that live alongside them. And what you can see from our data, which is the table below, is that the TT column has dramatically declined. So can you just show the next animation, please? Yeah, so there's been a significant reduction in the proportion of smallmouth salamanders that make up the salamander complex on Pelee Island, down from 14.84 to 3.8% of the population. And then we see the same sort of thing with the blue-spotted salamanders, a dramatic reduction, perhaps even a more extreme reduction in the proportion of blue-spotted salamanders on Pelee Island. And this is really worrying because the entire system hinges on the availability of smallmouth salamanders and blue-spotted salamanders, because the unisexuals that live there as well that are unique to Pelee Island are reproductively dependent on sperm that's provided by the smallmouth salamanders or the blue-spotted salamanders. So the entire system is really hinging on this 5%, which is appears to be in decline, which is pretty concerning. Next slide, please. One of the things that we've been doing to try and understand how we can help these animals is trying to understand the variables that impact the suitability of their breeding habitat and the places where the salamander larvae live. And so we studied some of the ponds on Pelee Island, including natural and constructed ponds. And what we found was that the presence of salamander larvae was strongly correlated with the amount of canopy cover and the presence of crayfish burrows. So maybe crayfish burrows are providing important terrestrial retreats for these salamanders. The catch per unit effort, which is like a measure of how many salamanders there are in the pond or relative abundance, was impacted by the amount of leaf litter in the substrate of these ponds and the presence of submergent vegetation, probably giving them good places to hide and providing food for the things that the salamander larvae eat. One of the key takeaways, though, was that the constructed ponds that currently exist on the island aren't really providing suitable habitat. They have lower canopy cover, they have less leaf litter in the substrate, warmer water, and fewer contain some submergent vegetation. And taken together, this means that the currently existing constructed ponds across the island aren't really providing the habitat needs for the salamanders. Ultimately, only about a third of the constructed ponds on Pelee, even though they were constructed largely to help amphibians, actually contained any salamander larvae. And then the ones that did actually have 4 to 10 times fewer salamander larvae compared to the natural wetlands. So these constructed ponds really aren't doing as good a job as maybe they could, or maybe we might expect them to do. Another key result that we found was that it takes these constructed ponds a really, really long time to naturalize. So ponds that are created will take sometimes 20, maybe 30 years before they reach a point where they're providing habitat comparable to a natural pond. So this is a little bit discouraging, but one of the things that I think is like a positive spin on this is that now we know the kinds of things that we can do to build these ponds better when we build new ponds, and also the things that we might be able to do to help those existing ponds naturalize a little bit faster. So if we can look at the next slide, we'll see a beautiful pond that was created by the Nature Conservancy of Canada. So as we heard from Jill, the Nature Conservancy of Canada has been busying away creating wetland habitat across the island. And we were in a position, because of the research that we've done, to be able to provide them some guidance and advice on how maybe we can build these ponds to increase their suitability for salamanders. So one of the things that was a takeaway from our research was that we need to build these ponds immediately adjacent to the tree line, which will help increase the canopy cover, also increase the input of this leaf litter into the substrate of these ponds. And then also we can do things to help the naturalization process and the closure of that canopy cover by doing things like planting trees along the margin. So you can see that the NCC has planted trees along the margin of these ponds. So this is an example of a pond that was built in, I think it was built in 2020, purposely built for salamanders. The size and depth were also things we were able to provide some advice on. And so one of the great and exciting results from our work monitoring this pond was that we actually found salamander larvae. So salamander larvae, even in the first year after construction, the salamanders had already found it and started using it, which is really, really exciting. Perhaps even more exciting was that we actually found that some of these larvae were surviving and metamorphosing and creating new salamanders. So we encircled these ponds to try and capture the emerging metamorphs. And so they come out of the pond and then they fall into one of our little traps. We check those traps twice a day, and then we remove the metamorphs and we release them after we document them and take a genetic sample. And so really exciting. For the last 2 years, we found that these ponds that the NCC has created are actually full of salamander larvae, and those salamanders are becoming metamorphosing salamanders and emerging from those ponds, which is really rewarding. And then this salamander here, this is the last thing I think I'll talk about, is just this is a beautiful animal. This is literally the first animal to ever emerge from that pond. So this pond was created in 2020, and this animal is the first salamander to ever emerge from that pond. And for me, this is a special animal because it's like a symbolic representation of the power of evidence-based conservation and the culmination of all of our work. You know, of course, we're continuing on this work to find out new and other ways that we can help this population and help the salamanders on Pelee. But to me, this animal is just the embodiment of the power of evidence-based conservation in action and collaboration with organizations like the NCC and also, of course, Ontario Nature. Thanks.
Thank you, Thomas. Great, so I'll move on to our last presentation by Ryan, who will talk about his research on blue racers across Pelee Island.
Awesome. Hi, everyone. Thanks for being here. Yeah, my name is Ryan. I've been working on Pelee doing snake conservation work and research specifically for blue racers for, well, I guess.
Ryan, so you're a little bit quiet.
Oh, let me. Is that better? Is that better?
Yes, that's better. Thank you.
Cool. Thanks. Yeah, so I've been doing snake conservation work and research specifically for blue racers for the past 5 years or so. And so I'm really excited to have the opportunity to share some of the information that we've gathered with you today. I'm going to be going through 3 different projects, so I'm going to have to go fairly quickly, but please feel free to ask as many questions as you want at the end. Thanks. Before I actually jump into talking about the research and results, I think it's important to contextualize everything by talking briefly about what blue racer habitat is. So because they're a species that needs to be hot, they occupy open habitats like alvars, grasslands, savannas, and they'll move along forest edges anywhere where they have lots of exposure to sunlight. But the problem with that is that those habitats, if they're not managed properly or have those disturbances like Smera was talking about, they quickly grow into thickets and eventually into woodlands or forests and become unsuitable for the snakes. And that's what's happening across a lot of the conservation lands on Pelee. And it's becoming a serious threat for the habitat loss of blue racer. So a lot of the places that are high quality habitat or were, like the picture here is a nice Alvar grassland that you can see. Actually, if you hit next, Melissa, it's transitioning into these thickets, and a lot of times it's monoculture of dogwood, and so those are becoming low quality habitats that the racers aren't able to use. Thankfully, some conservation work has begun where we're managing these properties, and that's awesome. And now we just have to keep going. Next. So when it comes to managing open habitats for snakes, there's really two main techniques that are used. One is the prescribed burn, and the other is mechanical removal of vegetation, where you do that manually. And so the first aspect of the research that I'm going to be talking about today is evaluating both of those tactics in their ability to create suitable habitat for blue racers. And so one way to assess how those are doing is through monitoring the use of each habitat actually by the snakes themselves. So next, we did surveys for snakes on the prescribed burn site, a mechanical removal site, and then 2 control sites for 1 or 2 years before the burn and the mechanical removal were implemented, and then for 3 years afterwards. So if you look at the graphs here, unfortunately, we had, like, first of all, one really high spike that was random in 2019 on the prescribed burn site where there's a lot of snakes. But unfortunately, over all the years in general, we did not see a difference in the number of snakes using the habitat. And in 2020, across all years, it's kind of an outlier. There was a really non-snaky year. There just wasn't a lot of snakes on the landscape across all the sites. And so if you hit next again, you'll notice that this control site also stays. Both control sites didn't have really any change as well, besides that 2020 year. But where we did have a change was in the mechanical removal site. And so before the mechanical removal was implemented, there was still a decent number of snakes at the site. In 2020 was a generally low year across all sites, so we can kind of treat that a little bit as an outlier. And then in 2021 and 2022, the number of snakes per survey increased quite a bit. Okay, next. So another method that I used to assess the effects of each of those management techniques on snake habitat was to use model snakes with little thermometers inside of them, and they recorded environmental temperatures as blue racers would experience them for 1 year before the management actions were done, and then 2 years following. Next. So if you look at these graphs, if you look at the dashed red lines, those represent temperature ranges where blue racers prefer to keep their body temperatures. And what these graphs show you is the average hourly temperatures that blue racers would experience over the whole summer in the listed habitat type. And what we want to see is the environmental temperatures, those continuous flowing graphs there, getting closer to the red dashed lines after the habitat management, since that would indicate a higher thermal quality of habitat. And that's simply because the more the temperature is closer to the snake's preferred temperature, without getting too hot, of course, because that can kill snakes, then the less time the snakes will have to actually spend sitting out in the open, thermoregulating, soaking up sun, where they're exposed to risks such as predators. So looking at the graphs, we can see that thermal quality increased the first year after the burn in 2021, but then it went back down in the second year in 2020. In 2020 went up, and in 2021 it went back down to below what it was before the burn. But the mechanical removal site, like the snake use of the habitat, increased in thermal quality for both years after the mechanical removal was implemented, which also suggests that it resulted in an increase of suitable habitat for blue racers. Next. Okay, moving on to another aspect of the research. We were doing road mortality surveys where a team of people were biking the roads of Pelee Island twice a day for 5 weeks in the spring and 2 to 3 weeks in the fall. And we recorded all reptile and amphibian observations that we saw on the roads. Next. When we map those observations, and this data is only from 2020 and 2021 so far, we see the road network on the island light up like a Christmas tree. But unfortunately, about 75% to even 90% of those points are reptiles and amphibians that were actually hit and killed on the roads. So not too many of them were found alive. And next. And what we're trying to get from this project is a map of highlights, a map that highlights road mortality hotspots, specifically for blue racers and fox snakes. And so if we look at the data points for snakes on the roads, we can see that they're kind of, hit next one more time. Thank you. We can see that a few hotspots are already coming up. And once these are fully identified with all of our data, it's our hope that we can use those to look at mitigation strategies that we could maybe implement in these areas. And some of those could be like flashing wildlife, road signs, or maybe some sort of information delivered on the ferry to tourists and visitors traveling to the island. And hopefully we'll decrease the number of snakes that die each year from the cars. And one thing to quickly note before we move on to the next slide is that a lot of these deaths are from neonates, so baby snakes that are born, and they're trying to cross roads from the nesting areas to go back into the natural habitat in conservation lands. And that's where most of our deaths come from. So the last project I'm going to be talking about is kind of the bulk of my master's research and has been my biggest endeavor so far, which was to produce a new population estimate for blue racers on Pelee Island. Knowing the size of a population is an important first step in identifying the conservation needs for a species. And the size of the blue racer population specifically is important for Pelee Island and for Canada because it's the only place in Canada where these snakes are left. And so the last population size that we had known was in 2002, when it was estimated that as few as 141 individuals were left, and that number was decreasing. However, the estimates produced at that time were presumed to not be completely representative of the entire island, as several areas that are now open for us to survey on conservation land were private lands back then, and the researchers couldn't get on them. Even so, it was hypothesized around that time that at the rate blue racers appeared to be declining, it was possible for them to be gone from Pelee Island by around now. But I've spent the majority of my springs and falls since 2020 on Pelee Island's conservation lands with my small army of snake researchers monitoring the blue racers. And I'm happy to say that I've been able to produce a new up-to-date population estimate for the snakes and that they're still here. So you can hit next. So I used a technique called the robust design to estimate population size. And I was able to do that for each of the seasons that we monitored the snakes. The first few estimates are a little all over the place with large margins of error, but that's to be expected because I didn't have a lot of data to work with, especially since we've been monitoring an endangered species that's very difficult to capture. That data, it takes a while to build up where the models can actually run properly and produce accurate estimates. But as you can see, as you move along the axis, the x-axis there, as more data is collected, the estimates become more accurate with lower margins of error. And you can see that the last two estimates all hover at around just over 130 individuals. And the average of all of the estimates put together is about 157 individuals. Now, it's completely appropriate to use either estimate, and actually there's more methods where you can use some of the middle estimates because the data kind of structures those to be the most strong. But I have to do a bit more research on that to back up my choice. So for now, I'm going to leave it at saying that the population estimate is somewhere between 132 and 157 individuals. And of course, with the margins of error as well, it could be slightly lower or higher. Okay, next. So as I mentioned before, estimates were produced in 2002 as well, but those estimates were for only 3 sites on Pelee, and so I can't compare the new estimate that I just showed you directly with that one, since I surveyed more areas than they did. So to look at trends in the population, I reran my analyses and excluded the data from my additional sites. And then I reran the 2002 data with the same software and produced population estimates for 3 different years. And so these estimates indicate that there has been a likely decline in the blue racer population at those 3 sites over the last 20 years. So the population went from approximately 167 individuals in 2002. That was the estimate I got with running it through the updated software. And it went down to about 112 in 2021, and then down to a mere 57 in this year, in 2022. And so I do think the estimate of 57 is a little bit too low. The data is getting kind of sparse, and so the accuracy of it lessens with that. But given our capture numbers across the 3 sites, we do have reason to believe that there was a large mortality event of racers at one of those 3 sites over this past winter of 2021 to 2022, which probably caused the bulk of this decline. And not only do these estimates show, reflect that, but we only had 4 individual blue racers, adult blue racers, come out of hibernation at that site this year, which is a 75% decline from our average number of captures over the previous 2 years. And we believe that this mortality event was due to a low snowfall and a harsh cold winter. Okay, next. So to end it off, I think one thing I want to say is, well, thank you to everyone, of course, but that population monitoring should continue for blue racers, especially at that site. They are still on the decline, and we're hoping that we can get some further trend analyses, and with all the conservation work that's going on, hopefully we can see those numbers increase. Thanks again.
Thank you, Ryan. So now Smera is just going to wrap it up for us before we start the Q&A portion.
Thanks, everyone. I'm sure we all learned a lot from all of the presentations today, and I see that there are a few questions. So before we move on to those, I just wanted to thank everyone who's been involved in this work. We're not even showcasing everyone that's doing work on Pelee today. There are so many partners involved. So just wanted to thank some of them on the next couple of slides. If you want to go next, Melissa. And of course, in addition to people actually completing the work on the ground, there are so many others involved who weren't presenting today. It wouldn't be possible to do all of this work without all the amazing field staff who are actually, out there on the hot summer days and on the cool mornings, rain, snow, no matter what the weather, they're out there collecting all this data so that we can draw some interesting conclusions. Next slide. And of course, a ton of research collaborators, student researchers involved, volunteers who've helped out with surveys, as well as data analysis as well. Of course, the Pelee Island Township and prescribed burn crew who were integral in ensuring the prescribed burn on the Stone Road Alvar property went smoothly. And of course, a huge thank you to our funders. Without their support, none of this work would have been possible. And just wanted to give a quick shout out. I'm not sure if some of you have seen through Ontario Nature's Instagram or YouTube channels that Pine Grove Productions has released 4 short videos profiling species at risk found on Pelee, and speaking with many of the people who presented today, as well as some other researchers and conservation partners on the island. So if you want to check them out, they're on red mulberry, the unisexual salamander complex, blue racers, and scarlet and mania. So again, you can view those on our YouTube channel or go to our Instagram account. The handles are listed below. And of course, if you want to learn more about the prescribed burn, we have an article that came out in our On Nature magazine in 2019. Oh, we have the wrong year on there. If you want to look at events for next year, a number of our partners in 2023, we'd love to have you out on the sites and see what we're doing and learn more. And of course, you can check out more on our Nature Reserves webpage on our website, and we'll be sending a follow-up email with emails for Ontario Nature as well as all of the speakers today. Next slide. So with that, we will move to the question period. We have about 8 minutes for questions, so we'll go through and answer some of these. So the first one, it looks like Ann Yagi, this one's for you regarding the hibernacula that you've been working on creating, specifically around what you meant by overburdening.
Yeah, so overburden is the soil, the topsoil on top of the bedrock. And it acts as an insulative layer to protect the bedrock from loss of thermal heat, loss of heat over winter. So when you lose that overburden, exposed bedrock, you can lower and cause more freezing within the fractured limestone area. And that's something that yeah, we have to be careful of.
Thanks, Anne. Okay, there's a question about the nesting sites. Oh, I think maybe that one might have been answered. There was one specifically for Jen on nesting requirements for fox snake versus blue racer, and if there's any research on that.
Yeah, so the answer to that question is that fox snakes also lay eggs, and they also lay eggs in similar types of nesting structures. So I was really focusing my talk on blue racer, but yeah, the nesting structures could also be used by fox snakes. And I'd be thrilled if they nested in them. And some of our more recent funding is specifically targeting fox snakes as well. So yeah, the nesting structures should be suitable for both species. And hopefully we'll get some nesting. Hopefully that answers the question.
Thank you, Jen. Okay. A question for Ryan about blue racers. There's actually a couple I think that could link together here. Were the blue racer numbers from the areas where the habitat declined from where the habitat has declined to thicket from Alvar? So basically, is there a change in the number of racers that we're seeing where the habitat has been restored? And then I guess another question about racers, since we have you here, is about possible captive breeding for racers in the future.
Yeah, so it's kind of hard to answer. So the problem is that the habitat encroachment, so that the Alvar is open grasslands and such becoming thickets, is island wide. So we don't have specific numbers that we can attest to it being exactly linked with those areas, but we do see the strongest areas of the population remaining as having more open land than others. And so back in like the 2000-2002 timeframe, where they identified a stronghold for racers, that's where we've seen the largest decline, and that's where we've seen probably the largest decline in available habitat as that area has become largely thicket as well. So it's definitely has an impact and it's linked, but we don't have the numbers that can specifically back it up yet. I'm going to be working on that in the future. As for captive breeding, it is possible. Scales Nature Park has captive bred racers in captivity from a US population they acquired a long time ago for many years, but whether or not that is actually going to happen for the blue racers in Ontario is unknown right now. It's in the recovery strategy to bring them onto the mainland once the Pelee Island population is stable. So once that is achieved, then those types of things will be looked at.
Okay, great. Thanks, Ryan. There's been quite a few comments and questions about getting involved in some of this work. And I just wanted to mention that we'll be sharing all of the presenters today, their contact information and Ontario Nature's contact information. And we'd love to have some of you join us for an event or specific surveys as well. So I just wanted to address that. Okay, I'm just trying to look through these questions. There's a question about whether there's been investigation into biological mechanical alvar restoration instead of prescribed burns. Specifically, this person asked about goats. So I don't know, Jill, if you want to jump in on this as a fellow land manager on Pelee, but when we've talked to members of the community, previously there was cattle grazing on the Sternard Alvar complex that maintained more of the open landscape before. And that's certainly a technique that's employed at other alvars in Ontario. There isn't actually livestock present on the island roaming free at this point. And there are some concerns with that too, because they can bring in a variety of invasive species. They're not very selective on what they graze on. They kind of graze on everything. And with the number of species at risk found on the property, or on alvars in general, they would really alter the habitat in a much more aggressive way than through natural disturbances like fire or selective mechanical shrub removal, which is basically just cutting back shrubs like dogwood, for example. I don't know, Jill, if you wanted to step in at all.
I think you covered a lot of the potential challenges with using livestock for managing these areas. Certainly it's, given that there's no livestock on the island, it's not something that's easy to experiment with. And there are a lot of potential concerns. So it would be a big project to try to make that happen. And I think at this point, the prescribed fire and mechanical removal are lower impact and easier to implement strategies right now for Pelee.
Yeah, thanks, Jill. Okay, so I think we're getting to the end here. If anyone has any further questions, like I mentioned, please do reach out to us after over email. We'd love to hear from you. And I will pass it back to Leslie to wrap up.
Hi, so thank you for that, everyone. Those were some wonderful presentations. I'm sure everyone enjoyed them as I did. There's a lot of great information. So thank you for all the work you do, and thank you for sharing that with us. There will be a follow-up email that will include the recording of this webinar, so you'll receive that in the next couple of days. And if you do have any other questions, the email for the Nature Reserves team is on this slide here. You can email Melissa and Smera there and ask any questions. And in the follow-up email, you'll receive the email for the panelists as well if you have questions that are directly for them. So thank you everybody very much for coming, and we hope to see you again soon. Bye-bye.